Okay, go right ahead.
Hello, welcome. Good morning to our BME seminar series hosted by the Department of Biomedical Engineering. Now, today we continue to offer this lecture in a hybrid format with both a virtual and live audience. The speaker will speak for approximately 40 minutes, so there will be 15 to 20 minutes for Q&A. Now, for the virtual audience, throughout the presentation, please use the Q&A feature at the bottom of your screen. It's important not to use the chat feature with the Q&A feature. We will try to answer as many questions as possible at the end. And of course, the live audience can also ask questions at that time. So now let us welcome Professor Rong Fan. Dr. Fan is a professor of biomedical engineering and pathology at Yale. He received a PhD in chemistry at Berkeley, did his postdoc at Caltech, and before joining the faculty of Yale in 2010. His current research interest is in developing single-cell and spatial omics technologies to study functional cellular heterogeneity and intercellular signaling in human cancers and the immune system. He has made seminal contributions to the field, particularly in spatial transcriptomics. He co-founded Isoplexis, Singleton Biotechnologies, and Agnes Atomics. He is the recipient of a number of awards, including the NSF Career Award and the Packard Fellowship for Science and Engineering. So welcome, Professor Fan.
Yeah, okay. So thank you so much, Ken, for the introduction, and thank you everyone for having me here and kind of getting me out of my tiny house and after this year and a half. And I almost feel this unreal, okay, seeing so many people here in the same room. And so what I'm gonna talk about today is, it's not just the spatial omics, so our latest research efforts in my laboratory, but a little bit kind of how I end up here as a biomedical engineer. I think many of you guys are very fortunate you have biomedical engineering major you can choose. When I was undergrad, no, okay. So I'm gonna kind of kick off my talk with this is my kind of disclaimer. And first of all, to really sort of say happy birthday to you guys. I think it's a 20th anniversary. Yeah, happy birthday to you. And so this is also the 20 years, I think, after I arrived in US. And so the reason why I'm able to come over here after this kind of year and a half stuck at home, largely because the advance of the science that made an amazing vaccine, all right, and I'm fully vaccinated, I'm able to come here. And so I'm gonna start my talk actually also from the story about vaccine about 15 years ago. And that was a very, very promising vaccine. I think everyone hoped that gonna work and complete cure. That's also a pandemic, that's AIDS, HIV. I think Merck developed the vaccine, this T-cell vaccine, and successfully pushed to on the way to the phase 3 clinical trial. And in phase 1, phase 2, all patients, all volunteers develop anticipated immune response, but still completely failed. And afterwards, the experts got together, discussed why. I think of many different reasons for sure. I think human monkey slightly different. They got to understand better. how the vaccine works in human, and rather than just in monkey. But one thing that got my attention is right here. So there's the assay, which is still the gold standard nowadays in the vaccine industry, is a kind of single parameter ELISpot assay to look at all different activated T cells. But biologists, immunologists back to that time already recognized that's not enough. Okay, you really need to look at a large panel of proteins secreted from single T cell to really understand the quality of that T cells. But unfortunately, no technologies exist, I think, to allow you to do so. And that time, I just switched my career from physical science to biomedical engineering to now really biomedical engineering, so kind of biomedical research. And I knew very little about biology or medical science at all. I thought, okay, this is something seems like we really need to have in the future for the vaccine development. Let me go ahead and develop this tool. And so, but you really need to look at the single cells in this case, so multiple proteins, because it is the capability of every cell to produce multiple cytokines. So if you have cytokine A and a B and a C produced by 3 different cells, that doesn't count. So single cell analysis back to 2006, that's basically, so very little about the single cell analysis you can do, very few technologies you can use. And so I think a few papers are read in early 2007. So if you can measure just a couple of proteins from one cell, that's a science paper, okay, published in 2007. And I think Nancy Albritton also was one of the pioneers, I think, working on single cell sort of electrophoresis using the analytical chemistry approach. But you don't get a throughput at all. You can just get a one cell and do electrophoresis. Literally, you are not able to do extremely high plex. You are not able to look at that many proteins you need to measure to understand the biology. And you are not able to measure hundreds or thousands of single cells in parallel. So I think what I proposed back to that time is, okay, can we miniaturize the DNA microarray, which I learned on the protein microarray, which I learned as the first thing when I joined Caltech Jim Heath lab. It's basically ELISA, but just a microarray-based ELISA. I was thinking, can we make the microarray much, much smaller and highly miniaturized, put in a tiny, teeny chamber. The volume is about 1 nanoliter. If you have one cell in 1 nanoliter chamber, I think you guys probably culture cells all the time, you know, okay, typically 1 million cells per ml. If I have 1 cell per 1 nanoliter, actually have the equivalent volume per cell. That means, okay, if that cell is producing the same amount of proteins, you should have the same concentration. So, okay, if that's the case, that should work if you can really miniaturize the microarray. So this is the microarray I proposed to do multiplexed protein analysis, in particular protein secretion, but actually you can also license the cell to look at intracellular phosphoproteomics as well. And so that time I actually reached out to Merck, BP, working on that clinical trial. But they said, okay, we completely discontinued the trial. We're not gonna kind of do anything further with those samples. But fortunately, I met Tony Ribas, and so Tony was working on cancer immunotherapy using CTLA-4, and later the famous PD-1 Keytruda trial. He started very first Keytruda trial. And I met with Tony, and we started to use this technology to look at TCR engineered T cells to treat human melanoma. I think the paper we published in 2011 demonstrated indeed that's totally true. Okay, if you can see polyfunctional T cells as predicted, and those polyfunctional T cells going to give you much better clinical outcome in patients. So in melanoma patients treated with the TCR engineered T cells, there's a MART1 engineered TCR adaptive transfer T cells. And so that's in the beginning. So people started to think about, can we look at hundreds or thousands of single cells, but also extremely high plex for proteins first, but people also try to do kind of different transcripts and other biological analytes. And so when I joined the Yale 2010, and I was very proud of this device I developed, Caltech, and I took this device to my clinical collaborator at Yale. And I still like this device, but they had no idea how to use this this beautiful device with that spaghetti stuff on the left. And then I realized actually this is an engineering problem. Since I'm in an engineering school now, I should take on that challenge. And so about like a half a year after I joined the Yale, I think Mario Roderer, who I reached out to even when I was a postdoc to discuss this T-cell polyfunctionality analysis, and he basically proposed on a theory polyfunctional T-cells correlates with the sort of immune durability and the potency and the durability in T-cell mediated immunity. And he invited me to a very nice conference in Greece. And so I don't know if anyone been there, you can recognize this is Mykonos. And at that conference, I was speaking back to back with Scott Tanner. And after Scott's talk, and Gary Nolan was there, and he was sitting in the second row of that conference room. And he thought, wow, that's transformative, okay? That's how come actually CyTOF started, okay? CyTOF started to be adopted by immunologists, and that's how the entire field kind of emerged, this sort of the high-plex proteomics single cell analysis. So after my talk, I was talking to Dirk Bush, and that was his response. Yeah, maybe. And I realized why, because the device is so complicated with those spaghetti stuff, and I really need to kind of sit down and think about how to redesign a device and really achieve high throughput and also high plex. And so Mario Roderer give me a list of all the proteins relevant to T-cell activation function, about 40 to 45 different proteins. But then I can measure all of them. I think that's extremely challenging. So over the about 5 years after I joined the Yale, and so I gradually kind of redesigned the whole device and applying the technology to different biological questions. I think not just the T cells, but also innate immune cells, as well as cancer cells. Actually, the cancer cells in the bone marrow, in the leukemic bone marrow, can produce many different cytokines to drive the inflammatory bone marrow microenvironment and then drive the pathogenesis. So, I think around the same time, I think my postdoc advisor Jim Heath also taking the technology a little bit to look at intracellular phosphoproteomics as well. So the technology is not limited to secreted proteins, but I think my focus in my own laboratories has been, how to characterize the immune cell activation states using the highly multiplexed protein secretion assay. And so if you are curious what the device looks like now, and this is much simpler. Okay, you don't have that spaghetti stuff anymore. And there's just a bunch of open microtroughs. If you know how to use pipette, you should be able to disperse the cells to the surface of that device and put a glass slide on top. The glass slide has a bunch of antibody stripes, and within each chamber, you have a whole set of antibody stripes to measure up to 42 different cytokines at a single cell level. So you can image which chamber has one cell, which has 2 or 3. So basically, you can look at single cell secretion, single cell proteomic profile, but also you can also see how when the 2 cells there, how they talk to each other. So if you can decipher the secret conversation between the cells by measuring the proteins produced by those 2 cells in the same chamber. And so the technology was used by, I think, both Novartis and Kai Pharma on the CAR T-cell therapy companies. Actually, Novartis reached out to us first. I just want to show you one example. And so I think the Kai Pharma use the single-cell proteomics technology we developed to characterize the pre-infusion CAR T cell products right before you give back to the patients. And they found indeed, okay, the ability for every CAR T cells to co-produce multiple cytokines as defined here as polyfunctional strength index correlates very well with the clinical outcome of those patients. All the non-responders, most of them have much lower PSI, polyfunctional strength index, at the beginning, even before you give the CAR T cells back to the patient. If you look at the responders versus non-responders, the p-value is statistically significant. So if you're an immunologist, you're going to look at many other things like whether or not those cells are producing interferon, and whether or not you have helper cells versus toxic T cells. But none of them give you no correlation with the clinical outcome except this polyfunctional strength index. I think the idea I realized actually pretty straightforward, pretty intuitive. So if you're asking whether or not the T cell is going to do the job, okay, then the T cells can hammer the T cells, give them the antigen-specific stimulation and see what they can do, right? So that's much more direct measurement of the T cell function rather than you look at all the profiles, okay? The analogy is just like for me speaking here, you look at my hair, look at my skin color, you say, okay, this is Asian from China, maybe he's not a good speaker, but why not put him on the stage, let him speak? Okay, the same thing, that's what I think. What's the best way to evaluate the T cell function here? So I think about a couple of years later, so that's another major breakthrough in the field. So not only in the proteins, you can look at transcriptome scale on the genes and a single cell level, but also extremely high throughput. So I think back to 2011, in my laboratory, I also started to work on the single cell transcriptome sequencing, but until 2015, I think the two papers published back to back to show you can actually molecular barcode on a transcript from the same cell and pull the CDA together to do pull the sequencing by computational reconstruct the single cell gene expression, then you'll finally achieve the extremely high throughput or massively parallel single cell transcriptome profiling. So the two papers published back to back 2015 from two different groups, but you find one person, I think, is shared in the authorship as Dave White. So basically, David developed all the droplet microfluidic systems. So the microfluidics really enabling this massive parallel single cell omics. So in my laboratory, we're able to combine, I'm not showing the protein data in this case, but we're able to combine the T cell protein data and the single cell transcriptome to really pinpoint the mechanism underlying this polyfunctional strength index or polyfunctionality of single CAR T cells. So we found something very, very striking. So immunologists know, okay, that the T cells can polarize into type 1, type 2, so Th1, Th2, some of them like Th17, but you should not see sort of the hybrid type 1/type 2, you may see that transient state. But we found that CAR T cells upon antigen-specific activation are most of the hybrid type 1, type 2 phenotype. That's very interesting. But on the other hand, think about those are the T cells made by scientists. Okay, they are not existing in sort of the natural immune system of the normal healthy individuals. They are made by scientists, engineers. So they're kind of like aliens or monsters. They can do something Charlie Janeway's textbook does not tell you. So, but on the other hand, this kind of confirms sort of those T cells, those CAR T cells are extremely polyfunctional. So the question is whether the polyfunctionality and the underlying mechanisms really dictate the clinical outcome. So this paper we show from just a healthy donor-derived CAR T cells, and then we try to ask whether or not the polyfunctionality of those CAR T cells really predicts the clinical response. So the clinical trial we participate in is so ALL. If you look at all the clinical trials of CAR T immunotherapy in AL, acute lymphoblastic leukemia, and chronic lymphoblastic leukemia and the non-Hodgkin's lymphoma. And so AL patients responded extremely well, almost like 90% of patients responded. But after a couple of years, okay, within 5 years, I think 50% of those responders developed the relapse. And so this collaboration with Penn, with Carl Jung, Joost Mellinghorst, and Steve Grubb, we look at all the sort of CAR T cells in the pre-infusion product across about 20 different patients. So we found indeed, okay, the responders, the relapse, okay, what is most interesting here is the relapse. So long story short, so of the relapse patients, they have the very potent type 1 response or type 1 function in their CAR T cells as manufactured, but they don't have the type 2 function. So it turns out type 2 function is really required to keep the patients under long-term remission. So that's a major finding here. It's very counterintuitive for most immunobiologists. So now we're validating this result in even much larger cohort, like 80 to 100 patients at Penn. So it's hard to do this experiment because we're doing like a 5-year follow-up of those patients. CAR T cell therapy relatively new. I think one of the first cohorts, I think treated by CAR T, that's about like 5 to 6 years now. Yeah, so I think 80, that's probably all the patients we can find to validate this result. So this is not limited to CAR T cells. Actually, we also look at tumor infiltrating T cells. In this case, we're not able to do sort of antigen-specific stimulation. We just kind of stimulate unspecifically or non-specifically. However, we observe exactly the same trends of the polyfunctional response T cells, as long as you have those T cells in the tumor, and then you give the immune checkpoint inhibitor therapy, those patients are going to respond extremely well. If you don't have the high quality T cells in the tumor, even though you give them immunotherapy drug, they still cannot respond. And so I think there's another way. So before treatment, you can predict the clinical response. I know in this field, people constantly look at mutational burden and hot versus cold, but we show actually the quality of those T cells in the tumor matters most as compared to all other matrix. So this is the diagram I used over and over, I think in the past 15 years when I wrote my NIH grant. So when you look at a tumor microenvironment, the T cells there and the tumor cells extremely heterogeneous, but that's still not a full story. Okay, you have them, but you have them in a well-defined spatial context. So I think Joanna wrote this review article or perspective article more than 15 years ago, showing the tumor vasculature and the immune infiltrates and the lymphocytes and the different tumor subtypes and the invasive versus the dormant, and the really form well-defined spatial interaction network. And so I think my former graduate student, I think who's in the audience right now, I think a postdoc with Professor Liang, and Yang did this beautiful work to look at the interaction between just two types of cells. The endothelial cells, in this case, you already have the microvasculature developed from the human endothelial cells, but you put in human glioblastoma tumor cells ex vivo from the patients into the microvasculature, say how they interact. And then we found, okay, that's very context or tumor subtype specific. And so all those different branches, if you are doing single cell sequencing, you know something called a monocall. You can look at a different differentiation trajectory. So this is what we show. So different differentiation trajectory, each dot here is a single cell. And for the sort of the tumor samples, that give you the highest co-localization with the microvasculature. Most of the cells are in this branch and a little bit in this branch, and the cells that do not care the microvasculature are in this branch. And then we know, okay, these two branches are basically proneuron tumors and also all the mesenchymal tumors, proneuron tumors are sort of the tumor stem progenitor cells. They're rare like perivascular niche. So mesenchymal tumor cells tend to migrate along the microvessels, but the classical tumor cells don't care. Okay, they just grow by themselves. So this is a perfect correlation, but you really don't know which tumor cells here interact with what endothelial cells, and so you still need sort of the spatial context. And this is a much simplified model, but the real tumor is much more complicated. So can we develop something to really map down the entire tumor in the spatial tissue context and also across entire genomes or transcriptome scale? And so if you're working on single cell sequencing and follow 10x Genomics website, you'll probably have seen this diagram over and over, and which describes the data structure for bulk RNA sequencing and the single cell sequencing and the spatial sequencing, right? So I'm stuck home kind of for a year and a half, but I still love travel. So I tend to use this analogy. So if you look at the map here, this is literally not a map. Okay, this is just in the shape of our country, but you know that's USA. And if you do single cell sequencing, so if you can get all the different states, and then maybe New York City in New York, as you get a cell subtypes there. But you just can't use this one plus this one to drive around, enjoy your trip, right? So you really need a full map with all the states, cities, and how they're connected, and to find the location you want to go and to enjoy your trip. So biology works exactly the same way. Okay, you know what cell types there, you know overall shape of the country. That's not enough. You really need a very detailed and a single cell level the spatial tissue context information. So over the past 5 to 10 years, I think the spatial omics technologies emerged to address this challenge. Can we really profile individual cells and directly in a tissue context? There are two major different technology directions or different categories. One is you can do imaging. Okay, people do fish for decades, but I was talking to Steve just a half an hour ago, and like a decade ago, you can do just a one gene, you do fish, and it's hard to do multiple. And you can do IHC, immunochemical staining for one protein markers, extremely to do even just a 2 or 3. But I was super impressed. I think in the past 5 to 10 years, I think folks in the field, in the microscopy imaging field, really pushed the boundary. And in particular for the SM fish that demonstrate you can get up to entire transcriptome scale, like a 10,000 genes mapped out, but that's extremely tedious and time-consuming experiment. However, that doesn't really give you the ability to scale and to get a high content and a large volume and also a large sample number. I really think the breakthrough came out when the spatial mapping was done with high-throughput sequencing. So basically, you can use the Illumina sequencing, but to reconstruct the spatial gene expression profile. So in this case, I think there are most of technologies are very, very similar. I'm gonna kind of walk you through very briefly the technology landscape in this NGS-based spatial transcriptome sequencing. And so the landmark paper came out 2016 when Professor Joachim Lamberg published this work in Science, showing you can use DNA microarray to capture messenger RNAs from the tissue section sitting on top of the DNA microarray. And then you making a cDNA on the microarray spots and then pull on the cDNA and do parent sequencing. And then you can computational reconstruct the gene expression profile because every single spot gives you a specific address code. So about 2 years later, I think 10x Genomics acquired that technology. And so 2019, about 2 years ago, that was the moment that the whole entire field exploded. And I think the slide-seq was published earlier that year, and then later it's the HDST, they use the beads instead of the DNA microarray spots to capture the cDNA, to capture the messenger RNA from the tissue. And 10x Genomics launched the first commercial product. And so we uploaded our paper about that time as well. And so of the next year, I think the technology, including the commercialized division, has been demonstrated for FPE tissue. And this year, this is even more exciting. I think 3 technologies came out to show you can get even like a 1 micron-ish spatial resolution by further shrinking the size of the spots. The 3 technologies called Stereo-seq and a 6-scope and a Pixel-seq. And so I think, however, all those technologies are more or less the same principle. I think coined by Professor Joachim Landberg. It's a surface barcode surface MRI capture for spatial transcriptome sequencing. So our approach kind of complete the opposite. So we don't take the messenger RNAs out of the tissue. Instead, we put the DNA barcodes into the tissue and do all the chemistry in the tissue matrix. And so we demonstrate even 2 years ago, we can do the entire transcriptome spatial mapping, but we can also map tens of proteins. So from the first spatial multi-omics. And this year we have sort of the spatial epigenome, we can look at not just the transcriptome or panel proteins and many other new opportunities that can be unlocked based on this approach called DBiT-seq. This paper was published last year. If you want to know the details, you can find out the cell paper, I think from November last year. What I want to show you is this device actually. So this turns out to be the simplest microfluidic device I ever worked with. So, all right. And all the inlets are pretty big. If you know how to use pipette, you can just pipette your reagents into those inlets. I think 50 different inlets. You don't need a complicated spaghetti fluidic tubing system. You just use the global vacuum. You can put the reagents into this regime where you have the tissue section. So this is really the simplest microfluidics I ever worked with. And so how that actually works is shown in this diagram. So you can take existing tissue on a glass slide. Okay, you don't have to bring your device to the microtom or to the cross-sectioning machine. You have existing tissue on the slide, and you put your microfluidic device on top of the tissue and introduce the first set of DNA barcodes, A2, A50, and each barcode, each oligo has the oligo DT to detect the messenger RNAs or the poly tail of the DNA antibody conjugates. And so afterwards, you do in-tissue cDNA synthesis. So you already afterwards, you already have cDNA in the tissue, but only one direction. You don't get a 2-directional address code, right? And then you remove the first device and put another device, microfluidic device on top with the microchannels perpendicular to the first flow direction to introduce the second set of DNA barcodes and then ligate them together. At the intersection, you have the combination of A and B. That combination gives you very unique spatial address code. And then you collect the cDNA, do parallel sequencing. you can computational reconstruct the spatial gene expression map. So if you play the trick with the DNA antibody conjugates, you can map down the proteins simultaneously in the same workflow. And so what I'm showing you here is the data we already published from the embryonic mouse brain. And so after this, the two microfluidic flow barcoding step, somehow kind of you press down the tissue, you can see a little bit kind of imprint into the tissue matrix, which is great because you can actually see where the tissue pixels are located. So the individual tiny, teeny squares. And you can look at all the messenger RNAs and all the proteins, in this case, on the 22. If you look at individual proteins, for example, right here, this pandothelial antigen, which tells you where the microvasculature and the brain microvasculature is located. I'm not pathology by training, just no way to tell where the microvasculature is. But from the sequence data, it's very clear. You can see the microvascular network. So epichem tells you that endothelial organs start to develop at this stage. But from the same sample, we have the whole transcriptome mapped out, pixel by pixel. And then you can do some sort of cluster analysis, very much like how you do your single cell RNA sequencing. You can find, in this case, 11 different clusters and also what genes determine each cluster. Just want to bring your attention again to this cluster, cluster 2. That looks like the network I just showed you before in the previous slide. And when you look at what genes are associated with this cluster, they are hemoglobin genes, they are red blood cells. So basically, those are the red blood cells stuck in the microvessels, okay? Biologically, maybe not so interesting, but I think this is kind of very cool. From different data, kind of proteins and the transcriptome, you can see how they really match each other so well. Okay, so we also apply much smaller tissue pixel size to map a specific region of the mouse brain. I would say random region because we didn't know much development about at that time. We just put our device right here to map down this area of the tissue. When I look at the image, my postdoc showed me, I thought, okay, you got a piece of tissue fiber from your king wife tissue, okay, not the mouse tissue. That happens very often if you work with microfluidics. And I said, okay, don't worry, let's still go ahead and get your sequencing data. It turns out that's not a piece of fiber, that's actual layer of cells defined by some markers, for example, PML, that's a melanocyte marker. So actually have a layer of melanocytes lining around some structure, that's the eye cell. Basically what you are seeing is the early stage the eye cell or eye vesicle development, and after just one day you begin to see the optical vesicle. So I think developmental biologists already know. So at this stage, the melanocyte, sort of melanocyte, epithelial melanocytes are just one layer of cells. And so what we really demonstrate here is the 10 micron pixel size DBiT-seq can resolve just a single layer of the cells directly in the tissue. So what is even more powerful in terms of computational data analysis we found is you can integrate the spatial tissue pixel data, the whole transcriptome data, and the single cell RNA sequence data in the same data set to perform unsupervised clustering. And afterwards, you see all the spatial pixels conform to the different clusters. And then you can do sort of your own gene ontology analysis for each cluster to find what cell types are associated with those clusters, and then go back to see where they are, right? So you can see spatially, many of those clusters are extremely distinct. And if you know the tissue pathology, tissue histology, you know what type of cells, what type of tissue associated with that cluster. And also, you can do another approach called label transfer. You integrate the single cell sequencing data and the transcript and a spatial transcriptome together, and single cell sequencing data well annotated, you know, on the different cell types. You just transfer the cell type or cell lineage label to the spatial expression, and then you can see where they are. I just show you a couple of examples. For example, some oligodendrocytes based on the single cell sequencing data, they are all oligodendrocytes. However, when we look at our pixels, spatial pixels, looks like there are two different subsets. And when you go to look at the spatial distribution, indeed, there are three different regions. Okay, they are all oligodendrocytes, but they are from different regions in the tissue. You can look at many other cell types as well, and they make perfect sense, like connective tissue. There's a ganglion neuron, and also you see some vessels, and also the red blood cells stuck in the vessel. And so whether or not the technology works for the FFP tissue, okay, if you really want to push the boundary to apply the technology to the archival clinical tissue specimens, that's a very important question. So we demonstrated that worked, I think, about a year ago. But recently we further demonstrated that works, but actually works just a perfect. I think the data quality is as good as the fresh frozen tissue. And this is a human lymphoma tissue. We mapped out the FFPE sample. You can see some clusters. I think the data is much more complicated. I just want to bring your attention to the quality of the data here. At the 10 micron pixel size level, we're able to get more than 2,500 genes per pixel and almost 10,000 transcripts per pixel. This is as good as the fresh frozen samples, which we're very, very happy about. And so also we apply this to human brain tissue. I think there's a collaboration with Professor Liang, and maybe later, I think Yang and Professor Liang can tell you a more exciting story from this, but we're so happy the technology works so well in this kind of post-modern archived human brain tissue samples. So I think everyone in this field got so excited earlier this year because Nature methods choose spatially resolve the transcriptomics as the method of the year. In the same issue, I think they also highlighted our work saying, oh, multi-omics goes spatial as well. And so, but what is beyond what we have demonstrated in that paper? So the leaders in the field, at least the sort of the industry leaders, they really believe with all the different spatial omics technologies, you're gonna see the next wave of biology revolution. And some people even believe you don't need a single cell sequence anymore in the future. That's what Joe Beechin really believes. Maybe I have slightly different opinion. I really think you need both. But I can't deny how much impact, how huge the impact will be once all those spatial technologies are broader applied to different biological and biomedical research problems. And so what we have further demonstrated in the past couple of months is, okay, number one, you can do proteins, but how many proteins you can do? And so I would say, I don't know. The answer is honest, I don't know, but I show you here up to 300 proteins are totally durable. Actually, we only use 10% of the sequencing depth to do that 300 proteins. In theory, probably can do even 3,000, but yeah, for a small lab, I think it's difficult to get so many different antibodies. So this is data from a human tonsil. And so you can see the spatial clusters from the protein data only. You can see very nice clusters that match the tissue histology and the tissue morphology where those B cell follicles are located. You'll see even a light zone and a dark zone. And from the same image, from the same tissue, we also sequence the whole transcriptome. We can perform unsupervised clustering of the whole transcriptome. I think the clusters in the spatial context match each other reasonably well. So you can also look at individual proteins on a B cell, for B cells or T cells, CD3, CD4, sort of in the T cell zone. those the B cell follicles. Also, you can look at the functional protein markers like IgG, IgM, IgD. And so, so 300, I'm not able to show you all of them, but just to give you a little bit idea how rich the data you can get out of this high-plex spatial protein profiling in conjunction with the whole transcriptome sequencing. So this is interesting data from the vaccine again. So we did this high-plex protein and a transcriptome co-sequencing, and from a skin biopsy sample. This is a post Moderna vaccine, I think the second dose, and we took a little bit of skin out of the vaccine site, vaccination site, and look at all the B cell, all the immune cells, and also the sort of the skin epidermal cells as well. I'm not gonna go into the details, but you just can see how beautiful this kind of spatial gene expression clustering and the spatial protein clustering. And so next, just a couple of minutes, I hope I can show you. This is way beyond just the transcriptome and a couple of proteins. You can look at the epigenetic organization of so directly in the tissue. So my postdoc Yan Shang, and he's an amazing engineer, but in this case, actually he figured out all the molecular biology stuff, and he's now teaching me the epigenetics. And so we apply the same cross-flow barcoding approach to look at chromatin accessibility, and instead of using oligo-DT to look at the transcript, to look at the messenger RNA, we use TM5 transposase to probe all the open chromatin loci. And afterwards, you insert the oligo to the open chromatin region, and then ligate the barcode A and ligate the barcode B, and then you have the spatial address code. So when you collect all the genomic DNA and the sequence, you can identify where they are in the tissue. And so what you are seeing here is the reference data from the ENCODE database, and this is from our aggregate sequencing data, but you can also see individual pixels from this is embryonic mouse liver. And so if you look at the entire mouse embryo, based on the spatial chromatin accessibility data, you already can do very nice clustering and to identify different tissue types. There's a liver, there's a spinal cord, but you see very fine, very complicated structure in the brain, because at that stage, I think the brain is the most well-developed. You see all many different neuronal subtypes and the brain tissue types over there. They all can be captured very well by the spatial chromatin accessibility mapping. So again, you can compare the data to the ENCODE reference data, the different organs from the embryonic mouse tissue, and they all match very well. But you see, we have much more granular information because we mapped out pixel by pixel rather than you dissect the entire organ to do the atoxic. And so again, we're playing the same trick to integrate the spatial atoxic data and the spatial and the single cell transcriptome sequencing. So using single cell transcriptome sequencing, you can get different cell types and the lineages, and then you see where the spatial pixels sort of co-localize with the cluster. You know, okay, that's the type of cell you have in that pixel, and then you can go back to see where those cell types are located. But keep in mind, what you are seeing here is not expression, but you are seeing the chromatin accessibility state. Okay, what I'm showing here, for example, the red blood cells, they're enriched in the liver. That makes perfect sense because at this stage, actually liver is the major hematopoiesis organ, not the bone marrow. And from using the same technology, we also look at a human tonsil again. And you can see on the B cell genes, T cell genes. But again, we're now seeing the gene expression, but actually we're seeing spatial and the chromatin accessibility state associated with different cell types, T cells, B cells. This is something I think no one else really can see. So something biological, super exciting, but no one can really imagine you can see at this level. Probably I should try to finish up very quickly. I just want to show you, we can further extend this to look at the specific chromatin modification rather than global chromatin accessibility. And so again, with my postdoc Yan Xiang, and he used the different antibody to target a different chromatin histone modification first, and then applying the transposer STM5. So very similar, but you just add the antibodies to target the specific histone modifications. So what you're seeing here is for 3 different histones and kind of several tissue sections from the same mouse embryo. And when you look at the data quality, it's extremely good. It's even better than the best single cell cut and tag data. And so if you compare the clustering, okay, based on the chromatin modification profile to the ENCODE reference data, they do match quite well. Okay, liver, there's a liver here, it's a heart, this is the heart. But again, you'll see much more granular information when you compare our data to the ENCODE reference data. But you know that those are several sections, right? You immediately get an idea. So different histone controls slightly different tissue development, different regions of the tissue. And you probably want to ask whether or not it's a multivalent histone control, histone modification control the development of, for example, the different brain tissue types. The answer is true. You already can see that. Probably you can do a better job if you can computational integrate them together. So this is sort of one of the histones we analyze. They just give you so better view of the different tissue regions and the different histone modification profiles analyzed. And so you can see the clustering based on sort of the global sequencing profile from this histone modification. You can also see individual genes predicted from the histone modification associated with different tissue regions. You can see very fine resolution for tiny features of the brain tissue as well. So I'm gonna kind of skip those slides, but very, very exciting data from adult mouse brain. We did a cut and tag to look at different histones controlling the cortical layer formation. But for the interest of time, I'm gonna skip those. But just kind of re-emphasize how exciting this field, I think, over the past couple of years, and what way further bring to the table is way beyond spatial transcriptome. And so how I think what the DBiT-seq is, I think I was really inspired by the speech given by our president last year, how he defined what is America. So it's possibilities. So I believe in the same thing. So DBiT-seq means unlimited possibilities. Okay, it's not another spatial transcriptome technology. I never believed it from the very beginning. So actually, that's a platform to develop many, many more spatial omics technologies to unlock new opportunities for biomedical research in the future. So I would say the limit is your imagination. I think you guys probably know better molecular biology. You can think of many, many more things you can do with this platform technology. So thank you very much for your attention. And yeah. If 2 seconds, okay, sorry. I was trying to say, if 2 seconds I have, I want to advertise. We do have the junior faculty position open this year in my department in the field of neuroengineering. If anyone ready to go and want to join Yale, yeah, send me email.
We have some time for some questions. We're going to try to use the microphone here so that everybody can hear us.
Hi, Dr. Finn. Thanks for your great talk. So I have two questions. The first one is, even though the pixel size is 10 micron, it could be still there are two cells sharing the same pixel, and both of their transcriptomic information will be collected. Is there a way to distinguish it? And also, if a cell is occupying 2 pixels, are you gonna organize the information afterwards during the analysis? And the second question is, so this very cool technique is for the fixed tissue. If we want to track the cell motion and then use the technique to map the cell motion and the spatial transcriptomic information, is there a future direction for the spatial temporal technique? Thank you.
Yeah, thanks. I think the first question is really the cell type decomposition. I think quite a few papers already published in this field to show how to do decomposition of either the Visium data or SLISEq data. Based on the same thing can be applied to our data. But I think that there's a much better way to do that. I think I think a different pipelines published right now, they just do sort of blind decomposition. So they don't look at what you actually have in each pixel. They just a computational kind of deconvolute the transcriptional profile in those pixels or spots. And that worked to some degree. But what we are trying to do now is really integrate the spatial imaging and the spatial transcriptional sequencing. So you can see, for example, in that pixel, okay, there's one T cell, one B cell, or one T cell, and even half B cell. So you can do much better the decomposition. So from the guide that supervise the decomposition, or use some machine learning to better refine the result of your decomposition or deconvolution. That's what I'm thinking. So it's totally durable with the current approach, but I think there are much better ways to do in the future. So the second question is the spatial temporal profile. Yes, I think a computational you can do so using something like RNA velocity, and then you look at a transient. So nascent RNA versus the more mature RNA and the splicing pattern, you can sort of do spatial RNA velocity, and we're trying that. I don't have the answer right now, but I really think that's durable. Yeah, but if you are talking about individual cells, you can watch how they actually move, how they actually differentiate. The data I'm now showing you, but actually collected that before all the tissue spatial mapping was. So you can do live cell imaging, how the cell move around or how the cells divide to give rise to and daughter cells. And then you fix them in situ or fix them on a slide, and then apply this cross-flow barcoding to sequence individual cells, but you know all the history, okay, before that snapshot, right? So you can go back and connect the history to your transcriptome data.
Dr. Fan, we have one question. We have time for one more question from the virtual audience here. This is from Louis Dingle. It says, excellent talk, Professor. One of the limitations with Visium for our group is that it has not been optimized in skin. Has DBiT-seq been applied to multiple tissues other than brain, in particular skin? If not, would you anticipate issues with its use in skin without further optimization? Many thanks.
Yeah, this is a great question. So whether or not the technology is kind of broader applicable to different tissue types, my short answer is yes. But on the other hand, I think I do believe, I think for different tissues, you need to do different optimization. So I think in my own laboratory, we haven't done all kinds of tissues yet. Probably 10x genomics can do so, but we have pretty good data from brain, I think a human and a mouse, and also skin and the lymph nodes and tonsil, and I think recently some data from I think beautiful data from skin, as I mentioned, and the liver as well. But the very difficult tissue to look at in the bone marrow, that's right on top of my my list to do spatial mapping. But of course, I think for different tissues, you need to, I think a tissue processing and optimization for spatial transcriptome, that's very, very important, equally important as compared to the assay itself.
All right, I think we need to wrap up.
Yeah. So thank you, Professor Fan. That is a wonderful, wonderful talk. Let's just one announcement. There will be a seminar 2 weeks from today for the benefit of the audience online. Speaker would be Professor Jim Well from Cincinnati. I think I was asked to announce that, and he will be speaking on that. So with that, let us thank our speaker again.